Bon dia a tothom. Buenos días a todos. Good morning, everyone. I want to welcome you to this seminar. My name is Javier Martínez Picado. I'm a research professor, ICREA research professor at the Institute of Age Research in Badalona. I'm also associate professor at the University of Vic. And it's a real pleasure to me to introduce a person that I work with very closely, Jakub Koinaki. He will be delivering this lecture today, this seminar, Seeing is Believing and Discovering How Do Scientists Use Light Microscopes to Study Viruses. And let me say a few words about Jakub, because I think when Jakub comes into place, it comes in three ways. He is a traveler. You'll see why. He is a great scientist, and also he is capable of simplifying very complex concepts, including physics and biology, into enjoyable seminars. So you'll see that through the present seminar. Jakub Koinaki completed his PhD at the Burnett Institute at the University of Melbourne in Australia. And that's why he said he's a big traveler, because he actually was trained far away from Barcelona, from Spain. In 2007, he moved to Heidelberg University, and this is Germany, as a postdoctoral fellow, where he worked on visualization of HIV substructures using super resolution microscopy. And he stayed there for almost 7 years. And then in 2013, he joined a laboratory at the Witherell Institute of Molecular Medicine at the University of Oxford in the UK, where his projects focus on mobility of lipids and proteins on the surface of individual HIV particles and at virus assembly sites, places where the virus do the budding. And now, since 2018, a couple of years ago, he is at the Is Research Institute, our institute here in Badalona, working on live cell imaging studies on HIV viral cell interactions, as well as establishing a new biosafety level 3 super resolution imaging facility. This is going to be, at this point, the only one in Spain where you are going to be able to do super resolution microscopy within a biosafety environment. And that has opened us new venues, not only in the field of retrovirus, which is the area that we mainly work, but also we initiated with Jakub projects on coronaviruses as well. And there are other groups working with him in other topics. So having said that, I hope you will enjoy his presentation. There are different parts, but at the end, if you have questions, please either write it down on the chat, or you'll have the opportunity to ask them directly to Jakub. So thank you, Jakub, and the floor is all yours.
Thank you very much. All right. Hello, everyone. I will share the screen, and then I will start with the presentation outline, what actually I'm going to do today. Sharing. Let's get started on this. I hope everyone sees the screen and me in the corner. So first of all, welcome. I hope you enjoyed the presentation, which I'll give today. And let me first of all outline of what how the presentation is going to be structured. So what I am going to do is I am going to first do a little 15 to 20 minute introduction on a little bit of theory of I will touch topics like virus size, architecture. I'm going to talk a little bit about principles of fluorescence microscopy. Then we're going to go over the basic anatomy of the microscope, which I'll actually describe a few parts of the microscopes which are important. Then I'm actually going to talk to you about the advances, what the technological advances have recently done to improve the state of microscopy, which allows us to study viruses in a new way, which was not possible before. And then finally, after these 15-20 minutes, we're going to go to the demonstration where actually I will have samples. And I am actually going to image some viruses, and I will describe everything as I go along the way. And hopefully we'll actually, you will see precisely in action how microscopists like me and other scientists are working towards utilizing the microscopy to find new things about viruses, or well, to discover, to uncover new things, and see the viruses as well, which is kind of cool, I think. And finally, I'm going to say a few words on conclusion after the practical part. I'm going to say a few words about the data that we acquired, and and provide some outlook for the future. So, with that, let's start a little bit about the very first concept, which I think is very important. Well, viruses are really small. That sounds really kind of banal, but it is, for us humans, I think it is very difficult to envisage exactly how small they are. Even with this slide here, which provides some sort of overview of sizes, it's very difficult for us to see. I mean, to imagine just how small those viruses are. I mean, here you have a comparison to a human here, and viruses are about 100 nanometers, 100 nanometers in size. And so well, the bottom line is they're extremely small, and it is it is very difficult for us to envisage exactly, well, what are we looking at, and therefore we need a very specialized equipment to see them, to study them. So, in terms of the size and architecture, well, classically, I'm going to talk about several different viruses. The one that I have studied a lot and has been studied by many people over the years is HIV. And HIV virus is about 140 nanometers in the diameter. It's 140 times 10 to the 9 meters. So overall, all viruses are about 100 nanometers in size. And without getting to too much detail, HIV and all the viruses are generally composed of lipid envelope, upon which you have envelope proteins, the surface proteins, and those proteins are important for virus attachment and fusion and being able to infect the target cells. And of course, different viruses have a different target cells. HIV targets cells of the immune system, SARS-CoV-2 targets the lung cells, and so on and so forth. And also, another very important thing, the virus inside have internal structures composed of many structural proteins, which allow it to perform different actions during the virus replication. And finally, the viruses have all the genetic material which allows it to replicate inside the cells, generate more copies of virus particles. Viruses come in different shapes and sizes. So, I mean, virus like HIV is 140 nanometers in diameter, more or less. There are some differences. No, the viruses are not out of a cookie cutter. They are a bit pleomorphic. There is a bit of a size variance. They are not perfectly in size. But something like, for example, Ebola virus particle, it's actually quite completely different. It is filamentous, up to 800 nanometers in length, and 80 nanometers in diameter. And then finally, SARS-CoV-2, which of course is on the news quite a lot. It is a significant development in 2020. SARS-CoV-2, so for information, is another virus which has a lipid envelope. Similar to HIV, has also virus proteins on the surface. This is so-called spike protein, and internal structures are composed of structure proteins and genetic material, and SARS-CoV-2 is around 100 nanometers in diameter. And that number of 100 nanometers, around 140 nanometers, is actually very important. Please keep that in mind for later, because actually this number relates to what microscopes, relating to the limits of what microscopes can see. And final point that I would like to make is, well, we visualize viruses as on the cartoons as very simple structures. You know, you can see genetic material, you can see a few proteins floating around. But if you look at the image on the top right, viruses are actually extremely complicated. This is a simulation of how HIV virus looks on the inside. And as you can see, unlike the cartoon, which shows a lot of empty spaces, viruses are packed full of proteins, and they are extremely complex organisms. And so no wonder the study of viruses is a complicated matter by itself when you just study viruses. But of course, then you have, if you want to study virus how does it infect the cell? That is also another massive layer of complexity. So bottom line is viruses are small, and they are actually very, very, very complex, despite the apparent simplicity that you see in cartoons. So how do we study viruses? And there is many different methods how scientists study viruses. And the one that I'm going to talk about, the one that I've been working for many years, is fluorescence microscopy. And it is an invaluable tool for scientific research. And I guess it is also very importantly because of the proverb that seeing is believing. That actually microscopes, fluorescence microscopes, allow us to look directly with our eyes and see what we're looking at. And thus, to me, at least personally, it makes it a little bit more real. A lot of scientific research is often a little bit abstract. You have tables, you have numbers, and those sometimes can feel a little bit abstract. But for me personally, the beauty of fluorescence microscopy is that you can actually see with your own eyes as you study viruses, cells, or other organisms. So that is actually, I think, one of the beauties of fluorescence microscopy. But in terms of what can fluorescence microscopy, what microscopy is good for? Well, first of all, like I was saying, it can give us some beautiful images. I mean, here on the right, you see cells that are stained in multiple colors, and that creates not only informative, but I think also quite a pretty images. What are the advantages of microscopy? Fluorescence microscopy as compared to other techniques like electron microscopy. Well, first of all, fluorescence microscopy is very gentle in general, and it is the preparation of the samples does not require any harsh chemical treatments. So therefore, if you're studying cells or viruses, you keep them in more or less physiological conditions. So there's always the danger when studying things is that you can change it by doing harsh chemical treatments. Whereas in general, in fluorescence microscopy, we do not affect the samples very much, and we study them more or less how they are in physiological conditions. This leads me to the second point. This allows for a potential of live microscopy. Unlike other techniques, you can study things as they happen in live. You can use model systems or tissues or entire animals, for example, to study things as they are in nature, without affecting the systems. Once again, without affecting those systems very much. And that is very important for scientific discovery, to study things as they are, rather than abstract and too abstract or harshly treated samples. Another advantage of microscopy is, as you can see on the image, is being able to tag specific things. And this is a very, very powerful mechanism, because we can focus only on the things that we're interested in here. So in the image of the cells, you can see that only very specific things are being labeled in different colors. You have a nuclei, you have cytoskeleton, you have a cell membrane, and we can specifically say we want to study, for example, a certain component of a cell membrane, certain component of a virus, and everything else is black and dark and does not actually interfere with our study. We can really focus on interaction between the things, which we tag, and they are fluorescent, whereas everything else is dark. And that is very, very important for studying things, because, as I said, viruses, cells are extremely complicated. And we want to, whereas if we want to just study a very small part of it, we do not want to all those other proteins, lipids, and glycans, for example, to interfere with our study. And finally, well, this is my perspective, that fluorescence microscopy is relatively simple and fast. The preparation of the samples is actually quite simple. I'm comparing here to some things like electron microscopy, which requires often quite, like I said, harsh treatments and so on and so forth. Whereas with fluorescence microscopy, especially, for example, if you do live microscopy, you grab the sample, you put it on the microscope, and you can start imaging straight away. So, for example, microscopy, like I was saying, can give us, not only beautiful cell images, but of course, the point about live microscopy. Here on the right, we can see immune cell in actual tissue, surveying the surroundings and trying to grab the antigens. So these are the advantages of fluorescence microscopy. Now, what are the disadvantages? I think a big disadvantage for many, many years of fluorescence microscopy was its limited resolution. And I will get to that point in a moment, because it's very, very important for the viruses. Because normally viruses were too small or to study via fluorescence microscopy. And what I mean by too small, it means that fluorescence microscopy cannot give you any information about the structure of the virus because of this resolution limit. And therefore, study of virus architecture structure has been pretty much always relegated to electron microscopy, which, while having some disadvantages, what it does, it allows you to study molecular details at great depth. And for example, here you have electron microscopy or electron tomography image of a single virus, HIV virus particles. And as you can see, you can recognize all sorts of details using electron microscopy. And that was not possible with fluorescence microscopy classically, but it is possible now, and I'll get to it in a moment. But first, I'd like to go over very briefly over several background concepts of fluorescence. What is microscopy? What is fluorescence? I think it's important for you to understand, the basics before I'm going to show the samples. So this is, of course, a very, very brief overview. Every area that I'm discussing, we can spend years and years to study it. So I'm just going to give you basic principles. So first of all, what is fluorescence? Fluorescence, in a nutshell, is effectively an excitation of atoms by light. In this case, excitation energy comes from a laser light, which is monochromatic, a single wavelength light. And what it does, it excites the electrons of atoms to a high energy level. And when energy levels go back to the basal state, to the resting state, then you have emission of the fluorescence photons, and effectively giving a fluorescence. So what does that mean in practice? You have several molecules. When you excite these molecules with excitation energy, and you have a graph here, with blue, you have excitation at wavelengths. You can see that molecules absorb certain wavelengths of light, the blue graph. And when those molecules absorb the photons with this specified wavelength, then they actually give off the fluorescence, which means the photons of the specified wave of several wavelengths given here with the red graph. And as you can see, that the excitation or absorption is always a little bit more, has a little bit of a shorter wavelength, and the fluorescence is always a little bit shifted to the right, so towards the more longer wavelengths, which are more red wavelength, because we're operating in visible spectrum. And why is that happening? Well, this is because when you excite atoms or fluorophores, some of the energy is lost, and the energy that is given back in the form of a fluorescent photon is always a little bit lower than the energy that you have provided. And this translates to a higher wavelength of the photons, which effectively means the photons of higher wavelength have lower energy. And this is called Stokes shift. So how this is useful to us? Well, scientists have utilized several different types of fluorescent molecules, and those generally can be classified into two forms. One of them are so-called fluorescent proteins, and maybe you have here the classic one is called the green fluorescent protein. And this protein, when excited with a wavelength around 480, 490 nanometers, is going to give you a 510, 520 nanometers fluorescence. So you can have proteins, which of course, proteins which are fluorescent. So you can utilize them by making, for example, cell or virus incorporate these proteins, and then you can tag them with fluorescence. However, the problem with those is that they are generally quite big, and some proteins, for example, do not like having an extra protein attachment, and they do not fold correctly. So another class, very popular class of fluorophores, fluorescent molecules, are so-called organic dyes. And those are small chemical compounds, which, as you can see, are very, very much simpler and smaller than a protein, and yet they are actually also very, very fluorescent. If anything, those compounds, like here, for example, I provided an example of a dye called Atto565. Those dyes have been engineered to provide extremely, extremely bright fluorescence. The advantage of those dyes is they are very much smaller, and so you can incorporate them into a greater variety of things. And they are brighter than a green fluorescent protein. So next question is, well, how do we get, for example, those dyes into the and tag various structure of interest, or for example, on a cell as well? Well, generally, scientists have been utilizing antibodies to tag molecules of interest. And as you know, antibodies are molecules from the immune system, and they're extremely specific to the given target or antigen. And scientists have took advantage of this extreme specificity of antibodies and started generating them against specific targets that we are interested in. So effectively, we took advantage of what immune systems of mammals are creating in general, and we took advantage of it and converted it to a tool to study molecules of interest in fluorescence microscopy. So effectively, what generally happens is you have a antigen or microorganism that you want to study, or a molecule that you want to study, or virus structure, for example, and you incubate it with antibody that is linked chemically to a fluorophore, to those organic dye that I was talking about. And basically, thanks to extreme specificity of antibody, it attaches only to specific structures and leaves everything else dark. And this way, thanks to attachment of antibodies to specific structures, we can take it to a microscope and study that structure that has been tagged fluorescently, utilizing a laser light and fluorescence excitation and emission, like I mentioned before. So this is a big basis of fluorescence. Microscopes. Well, microscopes have been in use especially quite a lot in use since 1980s, when the first confocal, or even in the early 1950s, when fluorescence microscopes started being popular. But I would give around 1970, 1980, when the microscopes were started being utilized routinely in laboratories. Nowadays, there is a lot of different types of microscopes. And here are different types of commercial microscopes that I have worked with. And what I just want to draw your attention to that these are actually, for example, commercial microscopes. And all of them have laser systems, oculars, and I'm going to go into the details about that. But just one of the things that you can see is that all of them have, so-called, if you can see on a camera, a microscope body with an ocular. And that is actually a common part of those microscopes. All those other equipment is detectors, lasers, control boxes. And those are important components. But I think this classically is what we could view as a microscope. But microscopes have there is a lot more to the microscope than just the ocular, for example, in the body. Another thing that I want to draw attention to is that commercial microscopes are not the only thing. For example, scientists also build their own microscopes. I mean, and that is actually a cool thing that I also do. Is in addition, if you want to, for example, create a microscope for your need that is very specialized in some particular way, you build your own. So rather than relying on commercial microscopes, you can see on the bottom, this is a picture from my former laboratory where we're building our own microscopes. And these days, I would view it like building Lego blocks. What you do is you grab optical components, you put them together to achieve certain effects. And here in the middle, I, for example, show a microscope that I was working with before, where there is, you can see a laser path, and utilizing a system of mirrors and lasers, we were directing those laser components to the objective, for example. And finally, custom microscopes can even come in form of Lego blocks. If you want to generate or create actually a quite competent microscope, you can actually build one yourself from Lego blocks. And there are actually quite a lot of projects that you can get yourself involved in and create your own Lego microscopes. So for example, one of them is so-called Lego-lish, and this is quite a competent quality microscope that has been created by scientists at IRB in Barcelona. And so, if you are interested in building your own microscopes or starting, there is a lot of resources online, and I encourage you to have a try. So, anatomy of a light path of a microscope. So, as you noticed, microscopes have this body in common. And how does it work? So let me describe first of all, how it looks in theory. What I show on a screen, I show you a basic microscope, and this is also the one very similar to the one that we have here. So generally, what is the light of? How does a microscope function? So first of all, you need the laser source or light source. I'm only going to discuss the fluorescence microscopy. Bright field microscopy is something a little bit different. But generally, you have the port where the fluorescent light is shining. And in this particular microscope, if you can see on the camera, the light which is generated here where the lasers are in this box is put via optical fiber and is fed into the microscope from here on the right. Then the light travels up to the objective. And objective is a very important part of a microscope where that laser light is focused onto the sample. Afterwards, the microscope, the same objective, collects the fluorescence. So basically, like I said, you excite the fluorophores of the sample, and then the same objective collects the fluorescent light of a different wavelength. And that light, when collected, travels down again and goes. We can either direct it to the ocular, or we can direct it back to the detectors. So here in this box on the right, we have both lasers, which generate the light for fluorescence excitation, and we also have detectors which obviously detect the fluorescent light. In addition, fluorescence microscopes often have an additional fluorescent light source. It's called it's a epifluorescence light source. I'm not going to go into the details, but that light allows us to very easily look at the samples via the ocular. Normally, when you use the lasers, you cannot utilize the ocular because of safety concerns. You don't want a laser light hitting your eyes. So we also have another light source which comes from the back and goes up, and then then goes into the ocular, which is gentler, and we can use it to study the samples directly before switching to the laser sources. Just one thing that I want to highlight is, well, why do we have oculars on the microscopes nowadays? Well, technically, like I showed on the custom microscopes, you actually don't even need that ocular. You do not need that body. All you need is an objective. You can have just a microscope stand where you put a sample, and you can look at everything on a computer. But I think coming back to the idea of seeing is believing, we still keep to that classical shape of the microscope with the ocular. And one of the reasons is that despite the advances in machine learning and artificial intelligence and algorithms, our human eyes, which are connected, which are part of a human brain, is still an extremely powerful tool for processing, imaging, and extracting information. So looking at things directly, I think, is still a very important part of microscopy, because our eyes can our brains can do processing that still the computers cannot. Of course, if you're doing something that is very specialized and you need to process a lot of data, the computers are really good at that. But our brains still are quite a lot better than computers in some of the tasks, and that's why we still retain on many microscopes those oculars for the first look. Where we can see the sample ourselves before we feed it to the computer, before we start analyzing the data on the computer. Okay, so I showed you a lot of different types of microscopes on the pictures, and this is to get a little bit more into details. This microscope is one of the types of microscopes that are available, and this one is called confocal laser scanning microscope. So first of all, what does confocal means? Confocal means in focus. Effectively, without getting to too much detail, it means that the sample that you, whatever that you see in the ocular or when you send the signal to detectors, is the only signal that you see is the one that is in focus. Any light that is generated by fluorescent sample that is outside of focus is discarded. And that is actually very important, because we only seeing any out of focus light causes the image degradation, or generally is troublesome. All that extra brightness comes from the not focused part of the sample, where we're not focusing, is generally problematic. And this microscope cuts that light away. Laser scanning microscope. So what does laser scanning microscope mean? So let's start with the sample that I show on the screen here. Let's say we have some virus particles that have been labeled. The little orange dye indicates that I took a virus particle, and I have labeled its envelope protein with a fluorescent molecule, and I want to observe those. So how does a laser scanning microscope work? It works very similarly to, well, I would consider like an old TV, where an electron beam was scanning across the screen. It is very much analogous to what is happening here. So what we're doing is we're scanning the excitation laser light line by line by line by line, and this excitation light then generates a readout. And we get a fluorescent signal here. You can see in the green. So generally, like I said, we're exciting in this case with a green light, and we're getting an orange light, fluorescent light out of the sample. And thus we get a final image after scanning. And this you will see is important because when I show this, show the actual sample, then you will see that the image is not built instantaneously. The image generally comes down as if it's built line by line. And final image, as you can see actually on this picture, is well, a bit blurry. The fluorescent image has a limit of resolution, and this comes back to the fact to what I was mentioning before, fluorescence resolution limits. So classically, unfortunately, fluorescence microscopy has always been limited in resolution. And these are the classical conventional laser scanning microscopes. And that limit is 200 nanometers in the focal plane, XY, if you prefer, and 600 nanometer along the optical axis. So whenever you have any object that you try to image, anything below this number, you cannot get any information about objects that are smaller than 250 nanometers in XY or 600 nanometers in Z. And there are many reasons for this. This actually predominantly is because of the wavelength nature of the light and also the imperfect ability of the microscopes to collect the fluorescence. But the bottom line is, if you look at the image at the bottom, is that if I have an image object of, let's say, hypothetical image object of approximately 40 nanometers, then that object, when viewed on a microscope, will always appear as a fluorescent point, a fluorescent blob of around 250 nanometers. And so, as you remember, I said that viruses are around 100 nanometers in size. So, unfortunately, if you want to get any information about the microscopy, do any study of virus structure via fluorescence microscopy, well, you cannot, because the viruses are smaller than the resolution limit of a classical microscope. However, over the last 10 years, there have been a revolution in fluorescence microscopy. For many years, people have thought how to bypass or cheat this resolution limit. And over the last 10 years, scientists have come up with several ideas on how to cheat this or bypass this resolution limit and create so-called super resolution microscopy techniques. And super resolution microscopy techniques come in many different nowadays, in many different sizes and flavors and types. All you see here is quite a lot of different acronyms. Do not worry about those. But what I want to highlight here is that for the last 10 years, people started coming up with a lot of different ways to overcome this limit. And this became converting to an extremely significant discovery in science, which has culminated in 2014 Nobel Prize awards to those three gentlemen over here on this, which I show on the screen. And they are the inventors of superposition microscopy. And for the contribution to science, they have been awarded a Nobel Prize in 2014, signifying how important the whole thing is. And so I am going to describe to you one of the superposition microscopy techniques, because the microscope that you have here in front of you is a compact superposition microscope. So it is a confocal laser scanning microscope, but it's also a super resolution microscope. And for more details, this microscope is called STED microscope, which stands for stimulated emission depletion microscopy. So in addition to your standard microscopy, it also incorporates inside a super resolution modality. And so how does this modality work? And I hope this animation that I created many years ago will help out to show you how exactly microscopy works and how superposition microscopy works. So let's have a look what happens in the microscope when you focus a laser beam on a sample. So here we have objective, and here we have a hypothetical sample. So the green cone represents laser light as is focused. And as I was saying, there are several physical limits to how we can focus light. Logically, by common sense, you would think that laser light can be focused to infinity. But unfortunately, that is not true. Due to the wave-like nature of the light, the focus of light is not perfect. At a certain point of time, fluorescent light photons start interfering with one another and create this, point of actual size, which is your focal spot is limited. And this focal spot is actually around 250 nanometers. And as you see, this is why we have a resolution limit in fluorescence microscopy. So here, those two red dots are hypothetical fluorescent points, and they are approximately 40 nanometers apart from each other. What happens when we try to use a laser scanning microscopy and to get information about the fluorescence of those two points? So this is the point, the focal spot of the microscope. And now let's scan the laser light across. As you can see, because of the not infinite ability to focus of the laser light, both of those points become fluorescent and start giving us the signal. So over here, when we look at the signal, you can see that we cannot so-called resolve information about the distance of those two fluorophores. For us, the signal, the fluorescent signal, appears as one. As one. We cannot get any information about the distance between those molecules. However, here is what happens when you use so-called, like I said, the STED microscopy. STED microscopy adds an additional laser in the shape of a donut, hence the donut in the presentation. And that donut has a zero intensity in the middle, and all it does is actually inhibit the fluorescence or inhibit the fluorescence of photons of anything that is within the point of excitation using that second laser, except that small zero in the middle. So when we scan the same two hypothetical points using a superposition microscopy, let's see what happens. And I'll pause here for a moment because, whoops, let me go back, unfortunately. Oops. Hi. What you saw was that, very interestingly, what normally would be a fluorescent molecule that is would be excited by excitation, the green beam, is inhibited by fluorescence excitation is inhibited by the STED laser, the second laser, except that small zero in the middle. And as we scan across, we get the signal which indicates that we have a separation between those two molecules. So all this, for just in a nutshell, all this superposition microscopy does is makes the region of excitation or the region of allowed excitation of fluorophores much, much thinner than in a classical microscope. And that increases the resolution of the microscopy. And in case of STED microscopy, this is about 40 nanometers, 30 to 40 nanometers. And that is a significant improvement over 250 nanometers and is below 100 nanometers, which is the size of the viruses. So with the superposition microscopes, nowadays we can study viruses in a way that could not be studied using fluorescence microscopy. So to put this all together, how do we study viruses in fluorescence microscopy? Superposition microscopy. So just to give you an overview, HIV has been studied, which is as you know, there has been for many years people working on HIV, and it is by far most studied pathogen by superposition microscopy. And people already over the last 5 to 10 years, thanks to the advance of into this technology, have taken advantage of this and have studied virus assembly, virus architecture, virus entry. And this slide, I just wanted to highlight to you, without going into too much detail, that scientists have already taken advantage of these superposition techniques and to learn quite a lot of new things, about very different parts of the virus replication cycle. And of course, this has a second additional effect, showcasing how useful this technique already is to other researchers in other fields to study viruses using these techniques. And of course, right now, SARS-CoV-2 is a very important pathogen, and so this technique is going to be very, very useful in studying SARS-CoV-2. So I will finish with this the theoretical part with a take-home message. Well, first of all, I hope that you realize that right now, the fluorescence microscopy is a very powerful tool of a scientific discovery, and also thanks to the advances in the technologies. Now, thanks to the breaking of the conventional resolution limit of microscopes, we can use these fluorescence microscopes and all those advantages that I mentioned earlier of fluorescence microscopy. But now we can study virus architecture. And so these techniques are now have become very, very useful to study virus architecture, virus cell interactions. So with this, I am going to now go into the practical part of the microscope of this session, and I'm going to demonstrate to you how we acquire several samples using this microscope. So first of all, I'd like to go over what samples are we going to do today. So, like you've seen possibly in the abstract, we are going to look at two types of samples. What I have prepared is a sample of HIV virus, and this is these virus particles have been labeled with VPR GFP protein. So this is the structural protein of the virus, and this has been labeled with in green. And what I have done is with antibodies, I have labeled envelope proteins, the surface proteins of the virus, and this will come up as an orange signal. Secondly, we have a SARS-CoV-2-like sample. What I'm saying like is because actually, as you realize, studying especially SARS-CoV-2 and also HIV is, it represents danger. And normally when you study a full viruses, you have to do it in a high-level biosafety laboratory if you want to work with a live virus. And actually, we do have, like Javier was saying, we do have actually a facility for studying those viruses. And right now, actually, my colleague Mari Carmen is, as we speak right now, working with a fully infectious SARS-CoV-2 virus, and is working on one of the projects that we have initiated in the Center for the Bioimaging here on the campus. However, of course, as much as I'd like, I cannot take a live fully infectious virus into this environment. So I had to do the next best thing and try to use so-called virus-like particles. And virus-like particles, and in general, they recapitulate part of the virus structure, but they are not infectious. And as you realize, exactly, scientists like to work on models sometimes because this makes it easier. I can study this model organism, model viruses. They're not infectious, and I can study them here. But if you want to work with fully infectious viruses, you have to do it in a higher biosafety environment. And to study those full infectious viruses, you need to have microscope in a biosafety environment. And this is exactly what we're going to do. This microscope is, much more easy accessible, but we're also going to have a microscope in this high-level biosafety environment where we can study fully infectious viruses. So what I have here in terms of a SARS-CoV-2 virus-like particle, this virus-like particle actually has a structural protein that is coming from HIV, and on top of it, it has a SARS-CoV-2 spike protein. So it only does not have any genetic material, it's not infectious, but it allows us to look at the behavior of that protein on the surface of this model. I can work with this here, and I can show it to you. Also, backtracking a little bit, this HIV sample that I'm showing you here is actually not fully infectious. It is fully virus, has all the protein, has all the, it is actually quite an advanced model of the HIV. It has all the proteins, genetic material, but some of the components of the viruses, small components of the virus have been disabled, and therefore it is, the virus is not infectious, cannot replicate in a human body, and therefore it's safe for me to work with it here. All right, so let's now put it on blank. And if you want to, what I'm going to do, I am going to now switch the screen, and I'm going to, hopefully you can see now the screen of the microscope. And this is the control panel of that microscope. And I'm going to start with the imaging of the samples and explain every step as I go along the way. All right, so we start with the HIV. I have several slides that I have prepared, and we have a microscope slide here. Hopefully you can see. Virus particles have been adhered to the cover slip surface, and it has been stained with the fluorescence with antibody, and we are going to put it on the microscope. So the first thing that you generally do is put oil on the sample. And why do we do that? I'm putting a droplet of oil on the sample is because the objectives that we are using for high magnification or high resolution are so-called oil objectives. And what that means is that that the contact between the sample, the glass of the sample, and the objective needs to be filled with oil, and this reduces the refractive index mismatch. So as light travels, there is less refractive index mismatch, and this allows you to get better quality images. All right, so I am going to now put the sample here. Hopefully, I do realize that you cannot see everything, but unfortunately, this is the limitation of this presentation. But hopefully, you see enough. All right, so I have put the sample, and I put it, I think, into focus. So now, my first step, what I'm going to do is I'm going to look down the ocular and try to make sure that the virus particles are in focus of the ocular. And then I'm going to switch to acquisition via lasers, and then you'll see the actual images as they are generated in my microscope. So I'm going to turn the load on. And I'm going very quickly to focus on the sample. Okay, there we go. Let me find a suitable area for imaging. Okay, let's focus here. All right, so switch it off. Now I'm going to put the laser light, the path of light to be acquired by the microscope. So hopefully you can see the screen. I'm not going to go into too much detail about what each component does, because that is a lesson on its own. But let's start with acquiring the overview. Okay, so as you can see, this is like I mentioned, this is laser scanning microscope. It continually scans the image. Okay. So let me change the color here, though, so I can. Okay, exactly. So green is the green fluorescence protein that I mentioned before, and this is the virus structure. And red, these are the envelope proteins on the surface of the individual virus particles. Those little dots that you see are actually virus particles, but we're looking at very, very big, small magnification. So let's stop and let's zoom in a little bit. So what I'm going to do is I'm going to zoom in the area, and let's continue acquiring. Oops, not yet. Let me actually go back to overview. And we grab a very, very small area, and let's start acquiring this. Okay, so this image might be a little bit messy, but what you're seeing here actually are, well, individual virus particles. So green, those green dots, each one of them represents individual virus. And red red channel, this is the signal that is coming from the envelope protein. When we put it on overlay, you can see that it's a little bit messy. As you can, so this is how confocal microscope, standard microscope would see this image. So now let's capture this image. Okay, so if you wanted to study a virus structure like an envelope protein, like I showed before, well, all you see here is, well, big fluorescent blobs, and you cannot really see any, well, you don't really have that much information aside from the fact that you know that the viruses are here because we have a green channel fluorescence, and we know that those viruses have envelope on the surface. But for example, like if you look here, look here, you can see that there is a bit of an overlay between the red, orange signal and green. So we know that probably this virus particle over here has envelope proteins on the surface. But how is this protein oriented? What is the detail? Well, we cannot find out about that using a standard microscopy. But as I was saying, coming back to my presentation, we have a super resolution microscopy. So let's see how it looks like when we activate the STED mode, the super resolution mode. So now we're going to acquire the image in confocal, the classical microscopy, and then in STED microscopy mode. So let's just click acquire, and the microscope will acquire first in confocal, and then in a super resolution mode. Okay, well, we can actually see quite nice things. So let's compare. Here, confocal image, you can see that there is a, let me make it a little bit brighter so you can see a little bit, the signal a little bit better. So here, for example, we'll have, I think, a single individual, not so bright virus particle. This looks like a small circle here. And we see over here a small red signal. That indicates that there is a virus, that there is envelope protein somewhere here. But it's very diffuse, and you cannot see, well, where it is exactly. But if we look in the STED microscope, you can actually see, let me see if I can zoom in a little bit. Actually, what I'm going to do is I'm going to go to the image gallery, and I think it's going to be easier for people to see a little bit. See if I can showcase this a little bit better. Okay, yep, there you go. So confocal image. Let me make it a little bit brighter again. Okay, you can see, well, there is a bit of an overlap. But when I switch to a STED microscope, you can actually see, make it a little bit brighter. Well, we can actually start seeing that there is a single molecule there, or there is very, very small. You can actually start localizing the virus envelope a little bit better. Let me try to find another area because I don't see anything that's super interesting in this particular image. But maybe, well, maybe here you have another virus particle where there is an overlap. We know there is envelope protein. But then if we switch into a STED mode, we can start seeing a little bit better what's actually happening here. Okay, there we go. That's a little bit better. You can actually see in this mode that there are two envelope proteins, or possibly two clusters of envelope proteins. Possibly this cluster represents one or two envelope proteins on each. So we can see that this particular virus particle appears to have two molecules side by side each other. Let me go over here. So there is a virus particle here, and there is one molecule, one cluster of ENV here, and a second cluster of ENV over here. And that already tells us a lot more about this particular single virus particle than with confocal microscope. Let me go back. I'm going to try to find another area. Maybe try something that has a little bit less crowded. So I'm going to move with the joystick, try to find. I can go faster. Try to find an area where there is a little bit less virus particles, because actually, when I generated this sample, I put a lot of virus on it. Okay, maybe this here. This area looks as it's not so crowded over here. Okay, so let me add some very, very small area over here. Okay, maybe this will be a little bit better. All right, let's have a look over here. So I'm going to repeat the same thing like I did. I'm going to try acquire both confocal and steady image. First of all, I'll just need to focus it a little bit. I'm using my mouse ball to zoom in a little bit, as in to focus the virus particles a bit more exactly here. So I'm moving the objective up and down a little bit to create to put the viruses in focus. All right, that looks fine. Let's turn on the steady mode and acquire the image in confocal and followed by a steady image. Okay, that looks quite all right. Let me just improve the steady image power by making the image one more time. Let's see if we can get a bit better image. Okay, yes, that looks a lot better, I think. All right, that actually looks much better, I think. So let's okay, here I think we're seeing quite a lot of interesting things. Let me see if I can put a magnified view. All right. So once again, please observe those two virus particles. I'll switch to select. So please observe those two virus particles over here. Both of them, there is one, if you look at green, there is two virus particles over here side by side. Both of them have envelope proteins. We can see that because there is a, in proximity, we have the orange signal here and orange signal over here. Well, from confocal microscopy, all we can say is, well, those viruses have envelope proteins on the surface. But if you look in a superposition microscopy, you can see that the arrangement of protein is very, very different. In this virus particle on the right, you have a single cluster of env proteins on the surface. So if you can imagine the virus particle is about here, that means that all of those env proteins are concentrated on a single side of the virus and are over here. But this is not the case on the virus over here. This virus, what appears to me, looks like it has we have a ring-like structure. And to me, this indicates that this virus particle most likely has env proteins quite diffuse across entire surface of the virus. So actually, this picture, you can actually start seeing the structural, the real structural details of the virus. You can start seeing a little ring, little donuts, which represents your individual virus particle. And I cannot overstate enough how big of a deal that is for scientists, moving from this into this. Now, using a fluorescence microscope, which is quite simple. I mean, all I did was put the sample and image it straight away. You can see with a click of the button, I have information about the structural details of the virus. Before, you had to prepare the sample in electron microscope. You had to have a massive electron microscope to do that. With this, we can study this with a click of the button. And like I said, another advantage is we can tag structures of interest, so we can now focus exactly on what we want to see. So I think this particular two viruses provided a very, very nice example of how we study study virus particles using these advanced microscopies. And as you can imagine, you can apply it to a lot more, to different viruses, to different structures, to cells, to organs, to many things. This is a tool of scientific discovery, and we here apply it to the viruses. Okay, so this was a HIV sample. I think this gives you a reference of what you can kind of expect from the virus particles in general. This is HIV. This is very well studied. Here I have the SARS-CoV-2 sample. And this is a little bit of a disclaimer. This is a very, very fresh sample. I stained it for the first time ever only 2 days ago. I had a very brief look at it. But to be honest, I'm still kind of curious what we're going to get out of it, because this is a fairly novel thing for us here. And as in this line of research has only recently started. And so I'm actually quite curious what we're going to get out of it. So let's move to next sample. So I'll take this one off. And what I'm going to do is call this sample number 2. All right, and I'm going to grab this one. So same procedure as before. I'm going to apply the oil on the sample. And let me do it here. Okay. All right. Like before, I'm going to try to first focus on the sample using the epifluorescent light. Okay, well, this sample is, well, not as great as the previous one. First of all, the concentration of the virus particles, those little green dots, is much, much lower. But let's see what we can get out of it. Let me find an area that has several of them, or quite a lot of them, if I can. Well, if not, then we're going to just have to try to get lucky a bit. Okay, I'm going to switch back to the laser line. We're going to send the signal to the detectors, and let's see what we find out. Okay. All right, so this is a little bit more difficult to see, perhaps. But yeah, those green spots, these are the virus, these are the SARS-CoV-2 VLPs. As you can see, this image is much, much less crowded. There is only very few of those dots. And let's see what we can find out here. Let me have a look at some areas of interest. All right, all right. Okay, let's maybe have a look here. Okay, so this is a little bit more messy, but let's see what we can find. All right. So here we are pretty much at at single molecule levels. This those diffuse signals in confocal. This represents that we're looking at very, very low concentration of an antibody that may have attached here by mistake, and we have 1, 2, 3 molecules of interest. Well, looking at this image right now is that well, the staining that I did in this particular sample is a little bit different. Is a little bit different. It doesn't utilize all antibodies. Actually, what I did here is I utilized protein called ACE2, which you know from the news and from your studies, and that it's actually this is a molecule that the spike protein of SARS-CoV-2 attaches to. I first attached ACE2 protein to the virus particles, and then I detected that ACE2 with the antibody. So this is a little bit more complicated way of staining, but it should still give me a fluorescent signal to in the point where the virus where the spike protein has been labeled. But looking at this sample, with those 3 virus particles, it looks like only one of them seems to have a signal, fairly bright signal associated with it. Okay, well, let's try to image this. This one's, as you can see, there is a bit of a signal here, but let's see what we get out in a super resolution mode. All right. Just acquire. Okay, this is like a big area. And not much here, not much here. Okay, well, this is a bit, as I said, this sample, I haven't looked very much at, so this is, I'm also particularly curious at what we're going to get out of it. So in confocal mode, we have a big diffuse signal. And in super resolution mode, let me actually crank it up later to try to make it as good as possible. Well, here we have what I would think, personally, I would think we're starting to see a little bit of a ring around it, a little bit of a ring. I would say there is several molecules of spike protein on the surface of this particular virus-like particle. yeah, so I think we're starting to see a little bit of a ring around it, indicating that yes, there are spike proteins on top of this particular one. Let me try to take an image again and see if we can get a little bit higher resolution out of this. So nothing here. No, not much better. Actually, a little bit better, I think. Let's see. Let me sharpen the image a little bit. Okay, we have at very low intensities, but the signal looks kind of specific. I think what we're starting to see here is that we can. I would guess that there are 3 small clusters of spike proteins on the surface of this particular particle. 1, 2, 3 in a sort of a roughly triangular organization. So this particular virus particle has a little bit of spike, whereas all others, well, do not have. Well, actually, so this already shows that this sample is quite different to the HIV one, where pretty much every single virus particle had an envelope on the surface, whereas this one's quite a lot. If you see a lot of green signals, do not have any orange signal associated with it. So this means 2 things: either, well, that not many of these virus particles have actually spike on them. This could be due to the problem with the model itself, or actually maybe there is a problem with the technique that I used to stain this image. So perhaps the well, not so much antibody, but I think what might be happening here is that the ACE2 protein is not attaching as well as I would hope it to the virus particles. What we see here is a lot of orange dots that are not at all localized with the green dots. That means I think there is a lot of ACE protein that is just lying on the cover slip and on a surface, and it's not well, it's not attaching at all to the virus particles. Let me see another area. I'll just do one more area, and then I think I'll start wrapping it up a bit in the interest of time, and see if we can find anything else. Okay, maybe let's have a look here. There looks like there is some sort of a localization here. I'm going to zoom in on this one. Well, not so much, actually. They're actually away from each other. Let me go back to overview. Let me try to find another one that might be co-localizing. If not, then, well, that's, well, this is a very, this is a new scientific sample. I myself do not know exactly what to expect exactly from it. Mm-hmm. Okay, maybe there is a single virus particle here. Let's have a look what we got. We can see out of it. Yeah, as you can see, it's moving. It's running away from us. It became detached from the. I think it's detached from the cover slip, and it's wobbling a little bit. So, well, it's no good, unfortunately. Yep. One more, and if not, then unfortunately, we'll have to call it a day. So yeah, most of the virus particles do not seem to have this spike protein on it, or we cannot detect any spike on it, which is a different thing, actually. Okay, well, maybe this one, maybe. Let me focus on it a little bit. Yep. All right, so we have a second one. Once again, big blobs of conventional microscopy, and let's see what we get when we do high resolution. Okay, well, this is interesting. And I think I've seen this yesterday when I very briefly looked at the sample. Here we have a virus particle, but the orange signal is a little bit of a distance away from it, What might actually happen is because the spike protein and AC proteins are quite big and antibodies are quite small, what might be happening here is that we are detecting fluorescence a little bit away from the actual virus particle because of the size of the spike protein and AC protein on top of that. So what might be, that's why we're actually starting to see a little bit of a distance between what I would consider a center of a virus particle, or I mean, probably the edge of the virus particle is about here, and the fluorescent signal. This would be my explanation. I'm not 100% sure, but that is possible. This indicates another important aspect of fluorescence superposition microscopy, that the size, how you label the samples, what is the size of the molecules that you use to label samples, start making a difference. In confocal, well, not so much of a difference. There is a big blob. You do not know exactly where the fluorescence is. In superposition microscopy, well, you can much better localize it, and therefore the size of the components does make a difference. All right, I think I'm going to stop here. And let's, okay, I'm not going to finish the session yet. I'll just leave it. I'm going to go back to the presentation, and let's do just a 2-3 minutes recap. So, result interpretation for HIV. If you can switch to the, well, you can see on the screen. So, what we have seen before in the standard confocal microscopy was a whole lot of green dots and red dots, which are the envelope protein. And we can only say that they are there. We do not know much about the localization or distribution of those proteins. In the superposition microscopy, as you saw, we were able to see that some of our individual virus particles have a small cluster of virus proteins, or some of them may have 2. We saw a molecule that has a ring on it, actually, which was very, very nice. And this already allows us to do statistics to get information in fluorescence microscopy about architecture of the virus. So, in a nutshell, we went from like if you look at the image at the bottom, we went from a big blob, blue circle is where the virus is, into this, giving us a whole lot more information about the virus particles using a fluorescence microscopy. In terms of SARS-CoV-2, this is one that I quickly grabbed yesterday. I mean, I only spent a very short amount of time on it. As you see, this is very similar to what we observed just a few minutes ago. In a confocal microscope, we will have just a not so bright image and also a diffuse image. But once again, the fluorescent signal for spike protein is a little bit away from what I would consider to be the virus particle itself. To me, it indicates that possibly the ACE protein, which probably attaches here, is creating this little bit of a distance, But, and unfortunately, another thing that was apparent from looking at this sample is that those virus particles, virus-like particles, have only very little spike on the surface. That, or ACE2 protein, is not as efficient as antibodies in creating this, binding, and therefore allowing us to visualize these spike proteins. That simply means we have to get back to the lab. We have to optimize the sample, see if I can incubate the ACE protein for longer, and see if I can get better images. So we, I think, already learned something new. This is actually useful information for me next time I'm going to prepare this sample. So we learned something interesting today. And just to finish off, the possibilities do not end here. I showed you something what I consider to be a very simple sample. It's just the virus particles. But then, for example, what if you add cells to the mixture? You have cells and viruses. So, for example, from the previous studies, you can, for example, look at how individual virus particles attach themselves to the cell. So this is a T cell. This is a HIV virus particle. And you can see how its envelope proteins are facing plasma membrane. In another example, you can study several different virus particles all at once. This is another study that I have been involved in. We looked at dendritic cells which engulf, eat different types of viruses. Here we looked at the distribution in this compartment of Ebola and HIV virus particles. And we used microscopy to accurately position in 3 dimensions the position of individual Ebola virus particles and individual HIV virus particles. And that is just static imaging. There is also a whole lot of live imaging. So, well, there is a lot of possibilities what you can do, combined with amazing technological advances. Fluorescence microscopy truly is a powerful tool for seeing and believing and scientific discovery. And with that, I am going to stop here. I first of all would like to thank Javier Martinez Picado. I am in his group for introducing me. I think he's unfortunately no longer with us, but I'd like to thank him for allowing me to work in his laboratory. I'd like to also acknowledge the groups of Nuria Izquierdo and Julia Blanco, from whom I've actually gotten the SARS-CoV-2 virus-like particles and ACE2 protein, respectively. And also, very importantly, I covered a lot of topics today, and I touched them only very, very briefly. I hope it still was understandable. But if you want to have any further reading, this article is a review that I've written in 2018, and it provides a broad overview of superposition microscopy techniques and how are those techniques applied to HIV virus, HIV studies. So if you want to, please have a look at it. Here is the QR code. And also, this second QR code, I provide a presentation link in PDF format if you'd like to have a look at it. I'll leave this here. And finally, if you want to look at that animation that I showed, it's on my YouTube channel. And if you want to ask me any question or contact me about microscopy, if you're curious about something, here's my email, and feel free to contact me. So with that, I'm going to stop, and let's see if there is any questions. Let's see if there is. Let me find the chat. I'm going to switch the camera off and stop sharing my screen. All right, if you can hear me, well, do you have any question? Actually, I'll continue sharing the screen in case you want to, if you want to have a look at that presentation. I'll keep it on the screen so you get the QR code. Mm-hmm.
Jakub,
yes.
Right now there are no questions.
Okay, well, sure. I went through a lot. I know that it's not easy to explain what a lot of different fields which are quite complicated in a single 1 hour and a half presentation. So I totally understand.
Well,
right.
Laura says, well, I don't have any specific question. I think you did a great job explaining the whole topic and everything was understandable. Thank you so much.
No problem at all. So I think we should leave it here then. And so once again, thank you for listening. I hope, well, I showed you something useful. I hope that, you might actually pursue this type of a science in the future. But if you have any questions, once again, please send me a message. I'm happy to tell you a little bit about microscope, about how to get into microscopy, about scientific career or whatever. So, well, yeah, with that, I think we'll finish. And thank you very much. And thank the organizers for letting me present.
Perfect. Thank you very much, Jakub.
Thank you.